Okay, in Chapter 7 here, we're going to talk a little bit about the actual molecular components of genetics, starting with DNA. We're going to talk a bit about the structure of DNA and how it is replicated. Again, here is the chapter outline for your perusal. So James Watson and Francis Crick were the kind of founding fathers of the structure of DNA. In 1953, they were credited with determining that the structure of DNA was this very notable double helix that we all know and love. However, because of the time, the 50s, women in science were not getting the same sort of notoriety and acknowledgments for their contributions. So Rosalind Franklin was a big-time player in discovering the structure of DNA. However, she was not given the credit, and only recently is she really getting credit for helping Watson and Crick determine the structure of DNA. So Crick, Franklin, and Watson were credited with discovering that DNA was in the form of this double helix structure, but there were some things that we knew about genetics before the structure of DNA, obviously. We knew that there was some sort of heritable component that was underlying these traits that were being passed down, and that came from Mendel, who was showing that there was these inheritance patterns when you did these specific crosses. And Darwin also theorized about these inheritable components when he was working with his theories of natural selection. And so there were things that we knew before the structure of DNA. DNA was just kind of a final piece that led to this kind of molecular revolution, but we knew kind of theories of genetics before the discovery of the structure of DNA. So even though we knew that there was some sort of hereditary component of living organisms like Mendel theorized, scientists didn't know exactly what was carrying that message, right? And so there was theories that it was the DNA, that it was RNA, that it was proteins, that it was polypeptides. Any sort of component of a living cell was theorized to be what was passing down this message. And so there's a classical experiment done with pneumonia, where there was this S strain of pneumonia that was really deadly to mice. And so if you inject this S strain into the mouse would die. However, there was an R strain that was a benign strain of pneumonia. And so if you were to inject that R strain into the mouse would live. It'd have no problem. And so this experiment would continue. If you boiled the pneumonia, then the mouse would live because you've killed those cells, right? However, now if you boiled the S strain and killed it, but also added that R strain that normally, when we looked up here, did not kill the mouse, now that combination killed the mouse. In addition, if you were to culture the blood of that mouse, you would get live S strain cells. So somehow those S strain pneumonia that he killed were magically appearing like they came back to life. So now the scientists conducting this research were tasked with figuring out, okay, what is it in that destroyed S strain, that boiled S strain, that is somehow bringing these cells or making those R strain cells turn virulent and making them kill the mouse. And so the first thing that they did was, again, they took that S strain extract and they put it with the R strain, and again the mouse dies, as we saw in the last slide. And you were able to recover live S strain from that dead mouse. Now they took that same component and they treated it chemically to destroy all the polysaccharides, so all the sugars and other polysaccharides from that cell extract, that boiled extract, are removed, and we leave everything else. And so when that is added to the R strain, the mouse dies again. So we've ruled out polysaccharides as being the component that gave those R strains that virulence factor, because even when we removed them, the mouse still dies. Now they move on to lipids, and they destroy all the lipids. They add that to the R strain. The R strain still kills the mouse, and live S strains recovered again. So lipids are ruled out. We do it with RNA, destroy the RNA, add it to the R strain, the mouse still dies. We destroy those proteins, add it to the R strain, the mouse still dies. And finally, they got to DNA. DNA was destroyed, and the rest of that S strain was added to the mouse, and the mouse lived. And not only did the mouse live, but the the S strain that is normally those R strains that get converted to these virulent S strains, it wasn't recovered. So DNA must have been what was causing this R strain to gain that virulence factor, that phenotype of being able to kill the mouse. And when you destroy it, it's not able to pass that to the R strain. So a second study with bacteriophages was used to further provide evidence that DNA was the inheritable factor that was being passed around. And so in this case, there was a two-part experiment. In the first part, they grew phages in a media containing sulfur-35. And sulfur-35 is a radioactive isotope of sulfur, so you can detect it with like a Geiger counter, or with radiosensitive film and things of that nature. So what they did is they grew it in this sulfur-35 media, and proteins have high sulfur contents in them. However, DNA does not have any sulfur in it. And so the DNA would be normal, not labeled radioactively, and the proteins were labeled radioactively. So they exposed these phages to bacteria and allow them to inject the bacteria. And then the phage ghosts release from the cell, and you have a bacteria that has DNA in it, and you have a group of phages that have no DNA in it anymore. They're just the proteins. And so when you look at these two components, you look at the bacteria that has the DNA in it now, and you look at the phages, and most of the radioactive isotopes are in the phages themselves. So that shows that none of the protein was passed to the bacteria. In the second experiment, they grew the bacteria in a phosphorus 32 media. And so this media is also radioactive and is similar to the sulfur 35. And so it's radio labeling the DNA. And there's not much phosphorus in these proteins. And so you're really only labeling the DNA and not the proteins or the polysaccharides of these phages. So these phages then go on to infect these bacteria, and the ghost phages, so the empty phages, are removed. And we see that most of the radioactivity is now inside the bacteria. So the bacteria that was not exposed previously is now highly radioactive. And so from that, we know that the DNA from these phages was being injected, and it was not the polysaccharides or the proteins of the phages. So this was kind of more evidence that DNA is actually what is responsible for carrying this message, and not proteins or polysaccharides or any other components of phages or cells. So some of this may be a little bit of review from your intro biology classes, or from high school for that matter. But the structure of DNA is it's made of 4 different nucleotides. We have the purine nucleotides, which are adenine and guanine, and then the pyrimidine nucleotides, which are cytosine and thymine. And so those are the 4 bases that make up DNA. And what's interesting about these bases is that different organisms have different amounts of these bases, right? So the A's and the T's, and the G's and the C's, they always have to be equal to each other. And so these numbers are a little bit off, and there's sometimes you have these transversions in DNA where you have an A paired with a G, and it hasn't been mismatch repaired yet. But most of these numbers are a little bit off because of the experimental measurement, but they should always be relatively equal. So we look at in the sea urchin sperm here, we have 32% that is adenine and 32% that's thymine. while we have about 18% guanine and 18% cytosine. So we should always have those two that are equaling one another. But the number of adenine does not have to equal the number of guanine. And so we'll get a little bit into some of these peculiar instances of things like codon usage bias, which we'll see in organisms where specific codons are used for specific amino acids, and it's kind of a conservation of energy type thing. But just note that the number of A's and T's are always equal, and the numbers of G's and C's are always equal in these organisms. So Rosalind Franklin's critical experimental results that contributed to DNA is that when Watson and Crick were talking about this in the structure of DNA, she had done these X-ray crystallography studies with DNA and found this interesting outcome that she showed to Watson and Crick, and this led to them discovering the structure as a double helix. And so it's really, this was kind of key evidence that they were looking for when they were trying to build this model of DNA. And so she never got the Nobel Prize like they did, and she kind of got left out of the limelight, although she's getting a lot more of the deserved credit for discovering or contributing to the discovery of DNA structure now, and rightfully so. So Watson and Crick took this information and some of the other information they had found from previous people. So those previous studies showing that it was DNA that was the heritable influence, and that A's and T's and G's and C's are always, equal to one another. And so they had all this, preliminary evidence as to what DNA is. And so they actually built really a physical model. It wasn't like they didn't have computers that were capable of doing graphical rendering back then. So they actually took the time to build this giant model of what DNA was, and really changed the way that genetics and all of biology was thought about from that point forward. So here's kind of an image. This should be very familiar to you. We have the double helix shape here. So we have these two spirals that are wrapped around each other in an anti-polar fashion. So they're going in opposite directions. And we have the sugar phosphate backbone that makes up the actual outsides of the ladder. And then the helix, or the actual lattices of the ladder, are those nucleotides that we talked about, A, T, G, and C. And so we have a monophosphate unit that is on the outside that makes up one of these backbones. And then inside we have these nucleotides. And so between A's and T's, there are 2 hydrogen bonds that form across here. And then between G's and C's, we have 3 hydrogen bonds that form across. So this comes into play when we're looking at things like if you're going to take a PCR class, the amount of energy required to break G's and C's apart, because there's 3 bonds as opposed to 2, is much greater. So A's and T's requiring or having only 2 hydrogen bonds come apart much more easily than G's and C's, which are stuck together a lot more difficult. One thing to be noted with the structure of DNA too is that, like I said, they're anti-parallel, or so they're going in opposite directions. And so I don't know how many of you have taken organic chemistry yet, but there is ways of numbering carbons in a structure. And so there is a 3' and a 5'. That's the number of the carbon in this phosphate backbone. And the 3' and the 5' are in opposite directions for the 2 strands. So if you're going 5 to 3 in one direction, the other strand is going 5 to 3 in the other direction, right? So when we start talking about transcription and translation, some of these machinery will read only in one direction. So they'll only read 3 to 5 or 5 to 3. So it's important to remember that there is polarity to these DNA molecules, in that some machinery, when we get to the actual replication and translation parts of, or transcription parts of the course, that this orientation of the DNA strands is important. So we know now that DNA is replicated in a semi-conservative fashion. And what that means is that the double helix will be unwound. So we have this replication fork here where the DNA is being pulled apart. So the two parts of this twisted ladder are being pulled to separate sides. And that exposes these nucleotides on the inside. And so when we say semi-conservative, during replication, we use this original strand, so in this case this blue strand, as the template to make this complementary strand. And so both of the two sides of this ladder are used as templates for new strands to replicate on. And so we say semi-conservative because even though if we were just to look at this part down here, it would look like we have two new strands of DNA. However, half of each of those two strands, so the blue half of each of the two, was originally already in another piece of DNA, a fully formed piece of DNA. So the only new piece of DNA are the gold paths. So it's semi-conservative where one half is previously been used and another half is new, has been generated from this replication process. So there were three alternative models for DNA replication before we kind of settled on and learned that it was semi-conservative replication that was causing, or that was the method for DNA replication. And so again, semi-conservative replication at the top here is where the two strands are pulled apart and one is used as a template for the matching strand. And so each of the two new strands have half of it that was part of that old strand. Conservative replication was an alternative theory, and that's where the original strand is red, or the two strands of the DNA double helix are red, and you make a fully complemented new strand of DNA that is 100% newly created, and there's no mixture of old DNA with newly replicated DNA. And then we have this dispersive replication, and that is a replication where different chunks of DNA are replicated and mixed with different chunks of new DNA and old DNA. So it's kind of this really bizarre mismatched theory of the pieces are made and then they're kind of put back together willy-nilly. So we're not going to focus too much on that theory. So similar to those previous bacteria and phage experiments, we could use radio-labeled isotopes again to determine what model of DNA replication is correct. So originally, DNA of this bacteria was cultivated in a media that contained those radio-labeled isotopes. So the first generation in our model, we have complete radio-labeled isotopes. Then that bacteria is removed from that radioactive environment, that radioactive media, and put with normal media. So during replication of that first generation, the original strands would be radio-labeled, but the replicated strands, because it's a new media, would not be radio-labeled. And then after those bacteria split and have offspring, the next generation would have those original parents that still have this radio-labeled strand, but then the offspring that used this gold strand as a template would not be radio-labeled at all, right? So if we were to look at this by doing centrifuging out the DNA, so those radio-labeled, those radioisotopes are heavier, right? Because they have an extra proton inside the nucleus, so they are heavier than the normal nitrogen isotopes. So in our first generation, we would expect to see all of them, all the DNA, have this heavy phenotype to it, right? Because that's all it was cultivated in. The second generation, we would expect to see 100% of our individuals be a little bit lighter because half of the nitrogen in those individuals is this normal N14 and then half of it is the heavier N15. So it'd be a little bit lighter than the first tube, but it would be heavier than non-radio-labeled DNA. But then in the second generation, we would expect to see two types of DNA, right? So we have this 50% radio-labeled, 50% not, and then we have completely non-labeled. And so in the test tubes, we'd have very light non-labeled DNA, and then this middle ground of DNA. And that's actually what we see. Now the predictions of the conservative model would be that, again, we set up the experiment in the same way, where we originally grow all of the bacteria in nitrogen media that is the heavy nitrogen, the N15. After the first generation, we would have individuals that are either heavy nitrogen up here or light nitrogen DNA. And then after the next generation, we'd have 1/4 that have heavy, because these two here are templates for the next generation. So we'd have 1/4 heavy and then 3/4 that are this light normal nitrogen. And so we would expect our test tube results to show that the first generation had all heavy, the second generation had equal parts light and heavy, and then the third generation would have 3/4 of them be light and then 1/4 be heavy. But although I've kind of buried the lead already by saying that this is actually what we observe up here at the top, so they proved that the conservative model was incorrect. Now lastly, this dispersive model, where we end up having the mixture of DNA, so it's 50%, but it's not on a strand by strand basis. It's kind of 50% chunks throughout. So again, they grew the original in that heavy media and then moved them to light media. So we'd still expect to see 50-50 in the second generation in this model, but then because it's not on a strand by strand basis, we would see 25% in each strand in the next generation, right? So here's how that experiment would play out. Like I said to start with, we'd have heavy DNA in the first generation, we'd have a middle ground of DNA in the second generation, and this is very similar to what we'd expect in the semi-conservative model, but the difference was that in the third generation, or the second generation of offspring, the third test tube, we would expect to see a halving of this first generation, right? So we would see 25% of the original radiolabeled DNA in each strand, but instead we get 2 bands because of the semi-conservative replication that we saw in the actual outcome of this experiment. So once Watson and Crick built this model, they also predicted that replication proceeded at what was called replication forks. And this was proven using these radiolabeled DNA, this time in bacteria. And so radiolabeled DNA, for the most part, would show that in bacteria we have this circle, right? And so these little dots are just the points at which these nitrogens were giving off radiation. But during replication, they noticed that there was kind of this weird bubble forming. And what that is the unwinding of this DNA to form this replication fork. And so we have one replication fork here, and we have one replication fork here, and it proceeds in this direction and unwinding this circle from both ends and allowing for semi-conservative replication of these branches as it proceeds to unwind. So as this double helix is being unwound, we have our replication fork right here in the middle, and the strand is unwound, and you end up with 2 strands that are anti-parallel to each other. Remember, we have a 5' end and a 3' end, and then the complement is in the opposite direction. We have a 3' end and a 5' end. So you have 2 strands, one a leading strand. The leading strand is the one where the fork is moving up the 5' end of it. So in this case, this bottom strand is 5 on this end, and then 3 down towards that end. And then you have a lagging strand, and the lagging strand is going to be the opposite of that. So we have a 3' here, and then as you can see, the 5' is way down on this end. So as this fork is being unwound, the replication machinery is in here doing all of its work. It's you know, adding bases and unwinding, and we'll get into actual proteins and their functions during this process in later chapters. But what's important is the direction of synthesis. So on the leading strand, DNA replication proceeds in that direction. So reading from 3 towards the 5' end. But on the lagging strand, the synthesis is in the opposite direction. So there's actually a movement of the machinery away from the replication fork where it's adding bases. And so this requires some additional finagling, so to speak, to allow for this replication to happen. So DNA polymerase III is a main component of this replication machinery that actually adds the bases to the template, or to the complementary strand, using the other strand as a template. But it can't start a chain of complementary DNA, it can only add to it. And so in order to actually, for polymerase to actually be able to add to a chain, there has to be kind of this addition, or this original piece of nucleic acid there that it can attach to kind of elongate the chain. And so that's where these RNA primers come in. So these RNA primers, primase is an enzyme that synthesizes these short RNAs, and they will bind temporarily to this lagging strand. And that allows DNA polymerase III to come in and start extending the chain in the opposite direction. And this RNA primer, or this primase that creates these primers, will make multiple primers as that chain's unwound and keep adding them, because DNA polymerase can only go a certain amount of distance before it loses its fidelity and doesn't, it stops working. So we see here that DNA polymerase will continue to add bases on the lagging strand away from the replication fork until it runs into another one of those RNA primers that was created by primase synthase. So once that DNA polymerase III runs into that RNA primer, a second polymerase, DNA polymerase I, will come in at the 5' end of the neighboring filled-in complementary strand, and will remove that primer and fill in the gaps. Finally, DNA ligase comes in, and although DNA polymerase I came in to fill in the gaps, there still needs to be kind of like a final pasting of the two strands together. So all the nucleotides are there, but there's a little nick in the DNA where where that nucleotides are the phosphate backbone, sugar phosphate backbone, isn't connected. So DNA ligase will come in and finish up and patch in that final little bit of backbone of this complementary strand, and you have a fully functional and fully formed complementary strand. What's amazing about this entire process is how high fidelity it is. So there's not a lot of mistakes made in the actual DNA replication. A lot of times these improper bases are from mutagens or problems with DNA damage and UV radiation or things like that that can change what type of base it is. But the actual the proofreading and the replication of DNA is very, very accurate. And that's because these DNA polymerases will work together, and they will actually proofread and make sure that the proper base is actually attached when this replication is occurring. So in this example here, we have our template strand here in blue, and we have a cytosine here, and so that should call for a guanine to be added. However, in our example here, erroneously, a adenine is added, and that's not the right match. Remember, A's goes with T's and G's go with C's. And so this same polymerase will proofread and say, okay, we added that, but it looks like it's not supposed to go together. And so remember, we had those 2 and 3 hydrogen bonds. Well, when you have that, the DNA kind of forms a little bubble where they don't want to go together, kind of like two magnets, right? They're kind of not properly attached. And so this proofreading will see that, and it will actually excise out that improper base and then allow for the proper base to come in. And so very, very accurate is this DNA replication process. Okay, so now we're going to point out some of these molecular mechanisms. We talked a little bit about these complexes or machines that I referred to in previous lectures and previous slides, and we're really going to start to get into detail about these molecular mechanisms for replication and transcription and translation and all these other things throughout the rest of the course. And so the first one I want to point out here is topoisomerase. And so topoisomerase is this enzyme that goes ahead of the replication machinery, and its job is to really remove those big kinks and rolls and secondary structures that the DNA is forming, and to kind of get it in line like a train track for the replication machinery. And then next we have what is called helicase. And so let me find my cursor here. So helicase is this little green enzyme here on the slide, and its job or role is to unwind the double helix. And so, not to be confused with topoisomerase, who removes like the big folds and rolls and loops that the DNA is making. Helicase actually pulls apart the two sides of that double helix. And behind that, we have primase. Now, primase we talked about a couple slides ago. Its job is to lay down those RNA primers required for DNA polymerase 3 to extend from. So DNA polymerase 3, remember, it cannot lay down fresh, it can't lay down on single strands. It has to have a double strand that it kind of starts and extends from. It can't just find a double or a single strand by itself and extend. And also remember that the DNA polymerase 3 always reads in this 3 to 5 direction, right? And so because of that, it can read fully this leading strand, but primase has to go onto that lagging strand and lay down those RNA primers for it to work on the lagging strand, because remember, that's going in the opposite direction. The two strands, one's 3 to 5 and one's 5 to 3. So moving on, we have DNA polymerase 3, and what that is the actual enzyme that lays those nucleotides onto the template strand. The beta clamp is just a clamp that kind of holds that polymerase 3 onto the strands, and so its job is to make sure it doesn't kind of move around, or it kind of just holds it there in this as part of this complex. So moving down, we have another one of those RNA primers. Remember, multiple primers are laid down, and then the DNA polymerase 3 will move until it reaches another primer on that lagging strand. We also have shown here these single-strand binding proteins, and the role, you'll notice there are all these little like ping pong balls, beer pong balls, right, that are coding these single strands. And the job of that is, when you break those hydrogen bonds are going to look for something to fill it, right? It's like a magnet looking to stick to something. And so when you pull apart the A's and the T's and the G's and the C's, then they're going to want to go right back together, right? And so the role of those single-strand binding proteins is to temporarily come in and act as their binding partner to allow those two strands to stay separated and not immediately go back to each other. And then as this DNA polymerase comes through, they get released once a new nucleotide, the complementary nucleotide, is added to that growing strand. So here we have DNA polymerase 1, and remember that fills in the gaps where that RNA primer used to be. And then lastly, we have ligase. This is a very good picture here where we have the entire strand, but there's a little nick in it that still needs to be filled. And then everything below this machinery is a perfectly good example of semi-conservatively replicated DNA. So here we have an example of DNA gyrase. So DNA gyrase is actually a subclass of those topoisomerases that we talked about in the last slide. So topoisomerase is actually kind of a category of enzymes that do this superstructural altering of DNA. And so gyrase is a type 2 isomerase, so there's a few different types. Regardless, just remember gyrase is a form of topoisomerase. And so gyrase, in this example here, we see that as this DNA fork is proceeding in the direction this way, we see that we have these kind of coils in our DNA. And so gyrase will go and it will cut the DNA and allow that, because there's tension in it, kind of like a spring, or this really dates me. You probably don't remember this, but when I was a kid, we had phones that had phone cords, and there's no wireless, wireless phones, let alone like, landline wireless phones, but people would have really, really long phone cords so you could kind of walk room to room or do stuff in the kitchen, and they get really, really tangled up. And so it would be a pain in the butt to try to unwind it because there's all this tension in it. So what DNA gyrase does is it cuts it, allows that tension to kind of unspool, and then allows it to go right back together. So that's how it gets away with, or that's the function it uses, I should say, to unwind these supercoils. So now we're going to talk a little bit about how this replication is initiated. So in prokaryotes, so remember prokaryotes being bacteria and cyanobacteria, etc. In prokaryotes, there are these points of replication called origins, and so that's where the replication forks start. And it begins by attaching to these DNA A boxes, and so these DNA A boxes are small 13 base pair sequences, and these little DNA proteins will come and bind there and start to unwind the double helix of the DNA and kind of pull it apart. And once that happens, the helicase, which in this case is this little green enzyme like we saw in the last figure, they're pretty consistent in their drawing of these enzymes. So helicase will come in where it made this little bubble, these DNA A proteins made a bubble, and it will climb onto this complementary strand, or this original template strand, I should say, and it will start to further unwind and pull apart this double helix. And at that point, as it's pulling it apart, we have two replication forks, right? So we have one on this side and then one on this side. And so the DNA polymerase III will come in behind this helicase and start to replicate in the two different directions of the replication fork. So remember, as we have this chromosome being pulled apart, the strand of DNA being pulled apart, we have two replication forks. So we started kind of in the middle here. This is our origin of replication. And we have one fork proceeding one way up the strand of DNA and one fork proceeding the other way up the strand of DNA. And so on that at that point, we have really two leading strands. So we have kind of this section here, which would be a leading strand, and then this section here, which would be a leading strand going towards the opposite replication fork. And then we would also have two lagging strands. So we have this top lagging strand here, which requires that primase to put those RNA primers in here, remember, because the polymerase III can only read in this 3 to 5 direction. And then we have a second lagging strand here. And with a lot of organisms, especially eukaryotes, there are a lot of origins of replication. If there's only a single origin of replication per chromosome, it would take a long time to get through an entire chromosome. And so there are multiple points of replication on one chromosome. So in this example, we have 3, but there are many, 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 many more than 3 per each chromosome. And so those replication origin points will create two forks that proceed out in two different directions, and they will grow until they meet one another, and we end with two fully replicated chromosomes that are replicated in a semi-conservative fashion. Okay, now let's look at the initiation of replication in eukaryotes, so humans, mammals, frogs, dogs, cats, etc. So initiation of replication is somewhat similar to prokaryotes in that there are these consensus sequences where these, or origins, where these proteins and complexes come in to start the whole process. And in this example, we have what's this thing called an ORC. So the ORC is the origin recognition complex, and it comes and it looks for these 11 base pair consensus sequences. Once it's there, it's kind of, that's the, laying down the target of where replication is going to start. At that point, these two other proteins come, and that's called CDC6 and CDT1. Those two proteins will bind to the now bound to DNA ORC, and they will recruit and help bring in helicase to kind of add to this machine, to start making this machine bigger and fully functional. Now once those helicase enzymes are bound to the DNA and start this unwinding process, CDC6 and CDT1, those proteins' jobs are done. They're just chaperones, so they just help get helicase in place near the ORC, and then they're out of there. And so after that, DNA polymerase III is recruited to that exposed strands that DNA helicase has now unwound, and replication can continue towards the replication forks. So one of the key differences between the genomes of eukaryotes and prokaryotes is that in eukaryotes we have linear chromosomes, so there's an actual end, as opposed to a prokaryote that has a circular chromosome. So what happens when we get to the end of one of these chromosomes? So at the very far 5' end of this linear chromosome, we have one of those RNA primers, but we're going to have a problem when it comes to filling in the spot left by that RNA primer. After that primer is degraded, in this case we have all four of the primers are degraded, we are left with this 3' overhang. And generally, as in the case in these first 3 gaps here, we would have DNA polymerase I come in and fill them in. But DNA polymerase I will only work if there's an upstream piece of double-stranded DNA that it can bind to kind of get on the train tracks, start going in and filling it in. And in the case of the end of the chromosome, we have nothing over here that allows DNA polymerase I to kind of get on track to move forward. So what ends up happening is after DNA polymerase I has come in and filled in the gaps and ligase has kind of filled in those nicks, we end up with this 3' overhang at the end of the chromosome. So the generation of this 3' overhang is a problem because it's exposed. So remember, there's all these little hydrogen bonds that these single-stranded nucleotides are trying to bind to. It's exposed to more damage and to unraveling, and generally the end of chromosomes are more susceptible to DNA damage anyway, and so there needs to be something to protect them. So what there is this enzyme called telomerase, and telomerase, what it does is it's a short RNA molecule that acts as a template, and it adds this it has this complementary sequence within the enzyme that will bind to this 3' overhang. And what it will do is it will add a little tiny sequence towards the end. So we've extended our 3' overhang, and then it will move down again because it's got a complementary sequence, so it'll kind of slide down and it will add to it again. So it'll continue to add to this 3' end to prevent it from degrading. There's a partner primase that partners with this telomerase that will come in and make a RNA primer that will be very far down on the extended 3' end of this overhang. And that will allow DNA polymerase to come in and fill in this gap that was previously there. And then ligase comes in and seals it. And so the normal end of our chromosome was this AACCC, which is right here. And we've extended the sequence, and this is not a gene, this is just what they used to call junk DNA. It's just kind of a non-coding piece of DNA whose only role is to protect this part from degrading and being pulled apart, and also allow the filling in of that 3' overhang. So from where our original replication started, where we had this AACCC end to the chromosome, we still have that, and we have a little bit of a buffer created to prevent damage to those actual coding sequences that are further into the chromosome. So finally, after the extension by these telomerase and primase of that 3' overhang, there is what we call the telomere. So the telomeres are these kind of protein-rich capped regions of the ends of the chromosome. You've probably heard something about telomere shortening and aging and things of that nature. We might discuss that a little bit in class. If you're interested, bring it up in class, and I can definitely talk about it. But at these telomeres, that 3' overhang, there are these proteins that come in, WRN, TRF1, and TRF2, and they kind of wind up that added extension of DNA, the non-coding extension or ends of these chromosomes, and they further protect it. So not only have we added extensions that are double-strand complemented, so they're kind of protected from those hydrogen bonds being exposed out in the open, but it kind of winds it up into a ball, and it protects these telomeres and this sequence of 3' overhang and telomere extended DNA from double-strand breaks and reactive oxygen species and things of that may damage the DNA. One further note on these telomeres, I said to ask you about it in class, but there's this interesting correlation between aging and telomere shortening, the length of your telomeres. So Werner syndrome is a syndrome where that telomere lengthening that we talked about is disrupted, and these people have much shorter telomeres because that telomerase doesn't come in and extends that 3' overhang. And so what ends up happening, like I said, they end up getting shortened and damaged. And what's interesting is that there is this phenotype of almost rapid aging associated with it. And so this is the same girl, and it's only a few years apart that these pictures were taken, but because of this syndrome, there's a lot of thought that aging as a product, or one of the products of aging, is shortening of telomeres, and that kind of manifests itself in this appearance of old age. If you're interested in any more in this, bring it up to me in class, and we'll talk about it a little more.